All right, so we went through transcription already, and it seemed like there weren't too many issues with this first part. So these are the answers. You can check your paper. And then also the first translation bit as well, with the mRNA codons and the tRNA anticodons. So this is the transcription and translation part. When you're translating between mRNA and tRNA, there is no thymine at all, so A is always pairing with U. And then we use this mRNA sequence to find the amino acid. So this is what we're going to use in the universal codon chart. Is what posted?
This, like, the answer key, I guess. Is it an answer key?
No, we're going through it right now.
Oh, okay.
We're going through the answers right now, yeah. So you can compare this with what you have on your paper, and I'm going to post this document and also this video of me going through it after we finish. So what organelle assists the tRNA in translating the mRNA sequence? It's the ribosome, the protein factory, which you may recall in Unit 2, we talked a lot about how the ribosomes were involved in protein synthesis, and this is why. Because protein synthesis is very important in genetics. So the tRNA is carrying the amino acids, and we're about to go through and use this universal codon chart. And that's really what I'm going to give you a little tutorial on. Please feel free to stop me and ask questions as we go through this. But it seemed like everyone was good up until this point, and then we started to get a little confused. So, yeah. Everything seemed good up until this point. Let's just discuss this number 3 for a moment. Is the tRNA anticodon more similar to DNA or RNA in nucleotide sequence? So you may have said that tRNA is more similar to the mRNA because they're both RNA, right? There is no thymine in tRNA or mRNA, but take a look at the strand of DNA compared to the strand of tRNA, and you might notice that they're basically exactly the same.
Wait, can I speak on this?
Yes, go ahead.
Yeah, when I was reading this, when I was doing the transfer, I realized the reason why there's 2 different messengers, the mRNA and then the tRNA, is because so the DNA can be the same at the end. Because when the mRNA takes it to the opposite, but the tRNA, the opposite of an opposite is the same, like 2 negatives. So then it ends up being the same, but the only difference is there's uracil instead of thymine.
Absolutely. And that just has to do with RNA in general does not include thymine as a base. It has uracil instead. And oh my gosh, actually, full stop. Speaking of that, I have a very hilarious TikTok that was shared with me by one of my students that is about this exact concept. So let's very quickly, I need to share this with you. I think you will enjoy it. A very small amount of profanity in this TikTok, but I think it's worth it.
Do you think it's easy for me to see you with somebody else?
You know what? Hey, you're the one who ended it, remember?
Yeah, because I was mad at you, not because I stopped loving you. But I've never felt.
You still love me?
No.
Sorry, I really felt like, especially that moment with uracil was really worth it.
Man, I just did that on camera. This is how I record. Anyway.
But yeah, definitely worth it. Definitely worth it.
Olivia Rodriguez's song, everybody during January made multiple TikToks over that song.
Yeah, I've played it many times in the Cafe B. People love it. People are fans. Driver's license, right? But okay, so the sequence of the tRNA is more similar to the DNA sequence. The only difference is uracil, and like Raphael explained, it is because the mRNA is based upon, yes, Joshua, yes. The mRNA is based upon that DNA template, but it is its complement. So then when the tRNA is created, it is complementary to the original mRNA, which makes it the same as the original strand of DNA, with the exception of uracil again. But if you compare the strands, you see they are almost exactly the same. The only difference is the thymine. So yeah, pretty cool stuff. All right, so as we continue, what amino acids did we find? So here's how we're gonna do this. So I'm gonna go through a tutorial. Oh, it's a three-way relationship? Okay, I like that. Yeah, I would say adenine is kind of two-timing thymine and uracil, right? She's with both at the same time. So, yep, yeah. So this is the famed universal codon chart, which looks more complicated than it actually is. So we're going to use the mRNA sequence. That's the only example, or excuse me, mistake that I saw anyone make. But these are the 3 mRNA sequences that we're going to use to find our amino acids. So here's how we read this chart. First, we take a look over here on the left-hand side, and we can see the first base. So for this first codon, we start with a C. So we're going to be looking in this row. So you see the first base is C, so our answer could only be in that row. Now you could just stare at the entire box until you see the 3 letters, but this is how you read it efficiently. Then the second base that we're looking for is U, so over here on the top, that's the second base, and we go here, it's only going to be in this column with the U. Are you guys with me so far?
That makes so much more sense because Rafa was reading the chart so easily. Because what I did was I looked in the entire chart and like scanned through which one it is instead of like doing the column thing. And Rafa got it in like 2 seconds. I was like, what's going on here? So
And
now
you
I finally
can
get it.
do that, Taz. That will work. It's just, it's not as efficient, right? It'll take you a long time, as you discovered. So that makes sense. So yeah, so now hopefully this helps you guys read it. And then the third base is over here, oops, on the right-hand side. Third base, and we can see here our third base, we're looking at C, U, U. So we go all the way over on this side, and bam. We found our codon, and we found our first amino acid sequence. Is that what you guys got? LEU
Yeah.
or leucine. All right, so now we're just gonna go through and find for the second amino acid sequence, or find the second amino acid rather in our sequence, GAG. So we're gonna do the same thing. First base, G. Second base, A. So now we've narrowed it down to this box. And third base, G again. GAG, and our second amino acid is glue or glutamic acid. Is that what you guys got?
Yeah.
Am I going too fast? Okay, Taz already put these all in the answers. Am I going, if I'm going too fast, we can spend another day on this topic tomorrow. So you thought it was a different word. Is that because you were using the tRNA? Because if you're using the tRNA, you'll get a similar amino acid sequence, but it'll be in a different order. You'll get glue-lu-sir instead of lu-glue-sir. So again, yeah. So if you're using the tRNA, you will get a similar answer, but it will not be correct. And part of the reason for that is because multiple codons, and you can see here in the chart, multiple codons, like here, for example, all 4 of these code for the same amino acid. And when we talk about mutations, we'll mention this again, but this is partially to give us insurance against mutations, because our DNA is getting replicated and transcribed and translated all of the time. So we have to make sure that there is some kind of an insurance against us having mutations. So if just one base is out of order, oftentimes that is a silent mutation, meaning it does not actually affect the amino acid or the final protein that is created. So, all right, let's go through and we're going to read this chart one more time. That is glutamic acid. And it's not really important that you know the names of the amino acids, just that you can use this chart. So now our third our third mRNA codon, what amino acid are we looking for? Let's find it. So our first base is U. Our second base is C. So now we've narrowed it down to this box. And our third base is A. So we're following this all the way over. Am I in the wrong box? No, I'm in the right box. To find our last amino acid, Ser or serine. Is that what you guys found? So then based on the little riddle that I provided you, you could write in this box LEUGLUSER, and that's totally correct, just with dashes in between it. But I gave you a little sort of key. If the amino acid LEUGLUSER, if that sequence codes for insulin, then that makes this protein that we code for insulin. And again, actual proteins would consist of more than just 3 amino acids, but for our practice purposes, this is insulin. All right, so now these are the answers to the last 3 questions. How many amino acids are coded for by the DNA? It's 3, because we have 3 triplets or 3 codons. So we could figure that out up here, 1, 2, 3, and we can see 1, 2, 3 amino acids. So that's just the number of codons that are in the sequence. It's 3. But you might have also interpreted that question to mean like how many amino acids are there, in which case there are 20 total amino acids that could be coded for by DNA. But it seemed like most people just got 3. Which protein does this DNA code for? Again, you could have just written LEUGLUSER, but the answer based upon that riddle is insulin. Now this one, I was going through with one of the groups right before we came out of the breakout room. So if instead of ACT, which was not actually the beginning of this DNA sequence, but we'll ignore that. The first DNA triplet was ACG, which amino acid would be coded for? So in order to figure out what amino acid would be coded for, first we need to do what with this DNA triplet? What do we need to do with that sequence of DNA to figure out what amino acid would be coded for? Yep, we need to
find.
Oh, go ahead, Raphael.
Well, I was, you just need to find the UGC in the amino, like the chart above.
Well, right, but I wrote UGC on here. Why? What am I doing? What is that UGC? What does that represent?
Oh yeah, you took the DNA triplet, which was ACG, and then made it RNA, which is UGC.
Right, so first we have to translate it to mRNA, and then take that UGC and go to the codon chart and find cysteine, or CYS. And then for the last question, we also have to do a little translation, sort of in reverse. What amino acid is carried by tRNA with the anticodon GUA? We're doing reverse translation, so we're taking that tRNA and turning it into mRNA. Because this chart is telling us what mRNA codons there are, and the answer is HIS. How do we feel?
Yes.
Most people like checking things off that they got right as we went through it? Oh, I'm so glad, Taz. That's such an improvement from when we started this assignment. I'm so glad. Raphael was a great professor.
Sarcasm.